Hello, and welcome to Ask Dr. Dawn. The opinions expressed in this program are those of the speakers, and this is a program intended for education and entertainment. It should not be construed as a substitute for a medical consultation. I am going to move right into an email that we did receive, and this one from Carl in Gulala, California. I'm a longtime listener and fan and had a follow-up question regarding your recent discussion of vitamin D. My wife and I were taking 5,000 units a day based on a number of recommendations, including yours, but then cut that to 500 IU every other day as a compromise when some other fairly trusted sources suggested that that might be high. I'm afraid I'll get whiplash via trying to follow on the various opinions on vitamin D supplementation. Well, my friend Carl, there's a lot more of whiplash in store for you, I'm afraid. For reference, my latest test result for vitamin D was 43 milligrams per ml. And I will tell you that that is a just fine level. I like it between 40 and 55, 60. If a person has autoimmune disease, I like it between 70 and 90. Cancer as well, I like to kick it up a bit higher. It has some anti-cancer effects. So my main question, going on with Carl's letter, regards the study that you described suggesting that people on statins didn't exhibit the longevity benefits from taking vitamin D if they were on statins. And this was a study from last week that looked at telomere length and showed that vitamin D had substantial effects at actually preserving the telomeres and preventing them from eroding. I believe it was a 3 or 4 year study, and during that time, people who were on, I think it was 2,500 units of vitamin D, did not show deterioration unless they were also taking a statin. There was a statin arm of the study where they were looking at other longevity benefits with statins, primarily benefits we would consider to be due to the anti-inflammatory, undisputed, by the way, and anti-inflammatory attribute of the statin. But it seemed as if the statin with the vitamin D might somehow interfere with the vitamin B benefit. And that actually could be through the fact that statins also prevent you from making CoQ10. And CoQ10 is a very important antioxidant, and it's especially important in mitochondria. So you could see how a putative or theoretical interaction, in my case I'm speculating, between having enough mitochondrial energy and having low redox levels in the mitochondria could have an impact and keep you from getting the benefit. Carl goes on to said I have borderline high LDL, and my doc is pretty good at avoiding synthetics unless necessary. He said he wouldn't require statins, but suggested I try taking red yeast rice, which, as I understand it, sort of mimics the effects of statins, but to a lesser degree. So my questions do you have any thoughts on whether red yeast rice, i.e. the phytosterols, this is also called red rice yeast in Carl's letter, might be contraindicated? And Carl, I'm concerned that it might do that it might prevent you from getting the effect. But that would be because red rice yeast or red yeast rice also interferes with the production of the ubiquinol or vitamin CoQ10. So what to do here? Well, first of all, I would get I would find out if I have large or small LDL particles. I would get an oxidized LDL, and I would see whether you really have a problem, because borderline high LDL is a very blunt instrument, and treating it with a statin is the kind of, well, it's what we have. It's like the old joke, when the only tool you have is a hammer. So we found that this is the only hammer that reduces cardiac death, which, as far as I'm concerned, goes to support the idea that this is an anti-inflammatory benefit. There's lots of other reasons to think that, but primarily, I mean, I can give you other drugs that will lower your cholesterol and lower your LDL, but you don't get a you don't not have fewer heart attacks 5 years later, even though your LDL is perfect. So it's the anti-inflammatory or the inflamed LDL that's the problem, and statins do that. So, any anti-inflammatory could potentially help reduce your chance of heart disease. For example, fish oil, something that's also got some substantial anti-cancer benefits. Like most nutraceuticals, they have multiple benefits. But I would say if you really want to be scientific about it, you could actually measure your telomeres. There's now a number of laboratories that will measure your telomeres for you. The tests range from about $100 to about $250. You could get a baseline telomere study and then take the vitamin D and or take the vitamin D and the red yeast rice if you've got an elevated LDL, small LDL, if you've got an elevated oxy LDL. And Quest runs these tests. These are not esoteric tests. You just have to know to ask for them. If you've really got the risk, then I'd say take both and monitor the telomeres and see what they do. If they're falling, then probably I would go off of the red yeast rice and check again, a year later and see if they've stabilized, because this benefit from the vitamin D was pretty substantially and significant. And, I really am encouraging people to follow through. By the way, I found out these telomeres. I simply searched telomere testing companies and got an AI overview, which I shall read to you. It says several companies offer this. Repeat Diex, LifeLength, and SpectraCell are prominent providers. And then it goes on to break down the various things, and with a little more fingers do the walking, you could probably find out the costs. So I'll leave it to you to navigate this jungle, and looking forward to hearing from you again, Carl. Straighten out my board here and move on to our first story, an important one about too many prescription drugs and how to do deprescribing. So what is deprescribing? Well, let me tell you a story. There's Argie. She's a widow. She's in her mid-70s. She's a former teacher, highly educated. She begins feeling bad. She's tired. Her stomach hurts. She gets a little agitated from time to time. She seems forgetful. So her children took her to a memory specialist, and the memory specialist tested her cognitive powers, thought she did pretty well. She probably could do the maze and do the draw a clock test. But he also looked at her prescription list. She was on 21 different prescription drugs. She was on some for diabetes, some for high blood pressure, some for her kidneys. And, they got her together with a clinical pharmacy specialist who got her down to 8 drugs, 8 from 21, and she's actually feeling better. Her cognitive, she's less nauseated, less confused, less agitated, more energetic, and steadier on her feet. A lot of people out there who are being diagnosed with dementia are actually drugged up. And I have seen that recently in my practice. I had one patient in particular come through my doors in the last couple of months. I've had several. This woman was on multiple types of the same drugs, several antidepressants, several antipsychotics, just way too many agents. And nobody wanted to take her off of them because she was convinced she really needed them. But I was pretty convinced after looking at that drug list that many of the problems that she came in with were actually, at least partially, side effects of these drugs. But it was not possible to deprescribe because I didn't have that patient's trust, and she wasn't ready. And you don't try to force people to do things in medicine. So we can hope that she will find herself in a controlled setting, possibly a hospital, where they will take a look at the list and not give them to her because she's in for, say, a surgery or perhaps a complication of surgery. And she'll have a couple of weeks of medical observation and a more the pharmacist in the hospital, who are very good at this because they often have to do it, will get her off many of these medications, and she'll leave feeling better than when she came in. It's very, very hard to do med reconciliation. It's very hard to get people to stop taking drugs. Imagine an 80-year-old woman. What are the chances that she's going to die in the next year? Well, they're not zero. If you change her medications and she dies, will you be blamed for causing her death by taking away a life-saving medication? Possibly. Who wants to feel that way? And especially since these drugs are generally being prescribed according to the standard of care. The problem is that you've got a cardiologist at Hospital A who's practicing standard of care cardiology, and a urologist at Hospital B who's practicing standard of care urology, and a rheumatologist at Hospital C who's prescribing standard of care rheumatology. But none of these doctors are completely aware of all the medications that the other doctors are prescribing, and none of them know about the over-the-counter pills, vitamins, and other supplements, all of which can interact. You can thank HIPAA, the Saint HIPAA, that is to say, the Health Improvement Health Information Portability and Privacy Act. Well, there's a lot more privacy than portability. In 1996, when this became the law of the land, by the way, I will add that it took them about 4 years to create this dog of legislation. And during that time, it slowed the development of electronic medical records. And when the electronic medical records did come out, they were siloed and they were legislatively protected. They were not required to be interoperable. Those of you who were voters in the early 1990s may remember Hillary Clinton's version of healthcare reform, which involved essentially what was then the technology, but it would have been nowadays a password-protected thumb drive that you carried around on your key ring that had all of your medications, all of your diagnostic information, the code number on your pacemaker, the 10,000, your drug allergies, your current pharmacy list, all of that on a little card that could be plopped into a card reader that would be standardized across our entire medical system. That was technologically achievable in 1992, and we didn't go there, all right? And the reason we didn't go there is because of, well, this was going to be a great cash cow if you could capture your audience. Very expensive to switch medical records, so once you picked one, you were likely to stick with it, even if you later discovered there were aspects of it you hated. There's essentially, you've got a captive audience because it's thousands of hours and thousands, maybe tens of thousands of dollars of staff time to move stuff over. And only large enterprises can afford to do that. So another thing that happened to me recently was I had a patient at the hospital, and I sent them to a skilled nursing facility. And it's not my first rodeo, folks. So a couple of days later, I went by the skilled nursing facility and checked that they were getting the medications I'd ordered the way I'd ordered them. And in fact, that was not happening. Instead of getting the medication 3 times a day, they were getting it once a day. And the 2 creams that they were getting were not being dosed properly either for their skin condition. And so we just, we had a little talk. We fixed it in the medical record. It was all very cordial. It's not their fault. Human error. When you have human error, you're going to get, guess what? Human error. And we had some there. Another problem is sometimes a lot of drugs have multiple different names. So a patient might be discharged with a prescription for Lasix to their pharmacy, or maybe they said, well, I'll send it to a different pharmacy. Now they're getting furosemide, which is generic Lasix, from their old pharmacy that doesn't know that they're also going to the new pharmacy where the doctor sent their antibiotics and getting their Lasix. So now they're taking a dangerous overdose of their medication, thinking that they're taking their medications correctly. So you've got to watch it. the more medications a person is on, especially if they start to become confused, they need to be taken off anything that can cause confusion. It's amazing how many other drugs there are that are out there that don't have central nervous system or cognitive effects. But we have to if we want the primary care doctors to do this, folks, you're going to have to pay us to actually do it. You're going to have a code and pay us for a half an hour of our time while we dig through these lists and go through these bags that people come in with. And ideally, this should be a service that you could purchase from your pharmacy and bring everything in and let the pharmacist, who is, after all, the expert on disease interactions, use their handy-dandy database to figure out what the concerns are and get back to your doctor and make the best decision possible for the patient. We need to fix this. And if you or your loved one is on more than 5 medications, I would strongly suggest you go into your doctor's office and say, which of these can I stop? Ask the question that way. Which of these, doc, do I really need? I think you'll get an interesting answer. We're moving into the mad scientist territory here. Many of you probably know that England is a bit of a stealth surveillance, well, not stealth, it's an overt surveillance state. National health is, in England, is going to, by 10 years from now, they've got a pilot program going. So in 10 years, every baby born in England will have a complete genome screening, a whole genome screening, which will assess the risk of hundreds of diseases. And that's every single newborn. Of course, that's a wonderful thing at one level. It will allow people to identify killer diseases. It'll allow people to get personalized health care. For example, we were just talking about drug interactions, and there are variations in how people process drugs, such that an educated physician could look at that and say, oh, you have a 2D6 mutation, ma'am. Tamoxifen won't work for your breast cancer because you need to have 2D6 in order to activate the drugs. So we'll just have to give you another agent to prevent a recurrence of your breast cancer. That isn't even done, that 2D6 testing, and it's a fairly common mutation. Many people are probably taking a drug that's giving them, reasonably bad side effects and not doing a darn thing for their recurrence risk. I worry about the surveillance state aspects of government genome testing to a population that is, by definition, at 5 days old, unable to give informed consent. And yes, it's helping patients receive personalized health care, and it would, in the long run, reduce costs by far more than it would cost, and help people live longer and healthier lives, avoid death from drug interactions. These are all really good things. But I have concerns about where this could go. People are so worried about vaccines containing chips that are going to be used to either track you or control you or whatever. And we carry our phones around, which clearly are tracking us and controlling us and whatever. But that's sort of different because it's voluntary. This would be involuntary, and it would be complete tracking with, I can't imagine how you could beat it in any kind of setting. Number to call, 831-900-5773, or you can send us an email to onair@ksqd.org. All right, in a nightmare scenario, we have this story. Cancer cells get a power boost by stealing mitochondria from nerves. It seems that cancer cells turbocharge themselves by actually sticking small little tubes into nerve cells and sucking out the mitochondria. Nerve cells have lots of mitochondria because nerve activity is very expensive energetically. This act of sneak thievery seems to give these cancer cells a boost that helps them survive when they metastasize. and spread to a distant tissue and don't have a good blood supply. So now we have a new culprit for what we can target to prevent metastasis. Researchers have all known that cancer co-ops the nervous system to fuel its growth, but we haven't really known how that works. Dr. Simon Grellett, a cancer neurobiologist at the University of South Alabama in Mobile, has been working in this area for quite some time. He and his colleagues work with mice that have a rodent version of aggressive breast cancer. They treated the mice with a chemical agent that blocks nerve function, and they saw that the metabolic activity in the cancer cells fell, confirming that the neurons are somehow influencing the tumor's metabolism. So growing in these same cells in a laboratory dish revealed that they were vampires. They extend a little tube from their bodies and poke it into the nerves surrounding them and suck out the nerve's mitochondria from the cytoplasm. Other experiments showed that these cancer cells with stolen mitochondria generate more energy than cancer cells without them. So the researchers wanted to see how these turbocharged cancer cells would fare during, well, let's just say that when you climb off of your home tumor and jump into the bloodstream, it's a pretty bumpy ride. So they pushed some cells through a needle and a syringe to simulate the buffeting that they would encounter in the bloodstream during a journey. Others they exposed to hydrogen peroxide to mimic the potentially damaging chemical environment they would meet when they climbed out of the bloodstream and found themselves in a foreign land. And the team found that the cells that had the opportunity to turbocharge were much more likely to survive than similar cancer cells lacking the extra mitochondria. So, yes, an explanation of a phenomenon, and yes, a potential target for reducing metastasis. Now, still in the mad scientist category, we're going to visit a bug called Entamoeba histolytica. It's a single-cell parasite that causes intestinal disease, but it can be invasive and attack cells and create liquefying abscesses. In other words, it appears to be digesting the tissue. UC Davis parasitologist Catherine Ralston has studied the genome of Entamoeba and now proposes a strategy to learn how it attacks human cells and evades the human response. So here's what we know so far. We know that it is usually an intestinal parasite and stays in the intestine, but something sometimes triggers a severe change, essentially turns on certain genetics within the parasite and causes it to begin chewing ulcers in the colon. From there, it can move to the liver, the brain, and the lungs and form these liquefied abscesses. And it'll basically kill anything when it's on the warpath like this, on a rampage. It'll kill anything you throw at it. It can even evade the immune system, and it can kill white blood cells. But how does it done? Well, all parasites are understudied, says Dr. Ralston, but E. histolytica is especially enigmatic. Just building the tools to study it has taken them years. But what they have found is fascinating. When E. histolytica enters the colon after a person ingests contaminated food, in the United States, it's most commonly seen in a person who recently took a trip overseas or immigrated from an area with poor water sanitation. When it invades an organ, if it goes on the rampage, it kills the cells in the invaded organs, but it doesn't stop to eat them. Instead, it wounds them. It basically tears a hole in them and leaves them to spill out their contents while it hurries on to kill other cells. And then it actually eats them. It takes bites out of the human cells. You can see little parts of the cells being broken up under a microscope. And they dyed some of these ingested some of the cells that they fed to the E. histolytica with fluorescent dye. And what they saw was that these cell fragments first accumulated inside the amoeba, and then the amoebas actually extruded uh, molecules from the cell onto their surface, like a hermit crab taking little bits of fragments of plants and putting it onto the shell that it's stolen from whatever animal was in it before. And they these proteins that they've put on the outer surface fool the human immune system. It prevents the amoeba from being coated with a class of molecules called complement proteins, whose job it is to find and flag invaders for destruction by white blood cells. So there's some other interesting fun facts about these camouflaged amoeba, one of which is that they have a very, very big genome, 5 times larger than that of Salmonella. And, well, that's a bacteria. And 2,500 times larger than HIV. But, of course, as we know, parasites are a more highly evolved, well, are debatably on the highly evolved, but definitely a more complex organism than bacteria. But E. histolytica uses a cellular process called RNA inhibition to control the expression of its genes. We're starting to use microRNA inhibition ourselves to prevent certain toxic proteins that a person might have inherited from being made in the body by giving them an injection that blocks the production. Well, it actually is the RNA that's the mirror image of what's coming off of the genome, and so it prevents it from being fed into the ribosome and keeps the protein from being made in the first place. But it turns out E. histolytica actually uses this as a control mechanism on its genes, and these researchers have put together a RNA inhibitory library, which allows them to drive the genome of the E. histolytica. I tell you, this stuff just gets crazier and crazier. So they are going to use this as a tool to identify which genes are required for the amoeba to bite the human cells or steal their proteins, and then see if they can make an microRNA, essentially, that would block that and disable, using CRISPR and gene editing. They might be able to come up with a way to block these parasites from being able to do their evil deeds, or come up with a drug that would block them that could be given. And certainly from a diagnostic standpoint, there's lots of potential here with this, so, particularly for subtyping the bugs so that the best possible therapeutic agent can be used. So everybody knows Tylenol, and most people know that the generic name for that is acetaminophen, but not everybody knows that that's not the only name, and that in Europe and a large part of Asia, acetaminophen is called paracetamol. Same chemical. The chemical itself, N-acetyl-para-aminophenol, is an excellent painkiller, unfortunately not an anti-inflammatory, but definitely an antipyretic, which means it brings down fever and it helps relieve pain. Currently, N-acetyl-para-aminophenol, or paracetamol, is being derived from petroleum, from fossil fuels. But researchers are working on a process that uses a more sustainable source molecule, something we have in abundance in the world right now: plastic waste. So they were able to transform a prolific environmental and societal waste into a globally important medication in a way that's completely impossible using chemistry alone or using biology alone, says co-author Stephen Wallace, a chemical biotechnologist at the University of Edinburgh in the UK. To convert plastic waste into paracetamol, researchers used conventional chemical methods to degrade and modify PET, that's polyethylene terephthalate, a plastic used in food packaging and textiles. It's got the little number 1 in the triangle, and turn it into a precursor molecule. They then add this molecule into a vat of E. coli cells, and something that in biochemical terms is called a Lawson rearrangement transforms it into into paracetamol. So they basically enzymes produced by these modified E. coli carried out the remaining synthetic steps to create paracetamol. When the researchers gave degraded peat to these bacteria, 92% of it was converted into paracetamol. So we're talking about straw into gold. We're talking about garbage into drugs. And actually a drug in overdose, I have some problems with it. And certainly if you are a regular drinker, you should steer clear of it because there's a toxic intermediate that will definitely accumulate if you are a drinker, and they both can injure the liver quite a bit. So it's not good to take a couple of Tylenol for your hangover with a beer chaser. But you probably knew that. Now, moving on to the brain, and you may have seen this already. A new study coming out of MIT suggests that relying on AI for writing reduces brain engagement, and this was a very interesting study. The researchers used EEGs of the brain, linguistic analysis, post-task interviews, and they found that using ChatGPT weakened participants' neural conductivity, their memory, their sense of ownership over their writing. And while the AI allowed the tasks to feel easier, this lack of effort could, over time, dull cognition, critical thinking, and creativity. So we have to be careful how we use these new tools, just like when I've been going to Bali for 30 years now, over 30 years. And when I first went there, when you would go around in your Jeep with your driver, there would be a lot of people just walking. And you would see people on motorbikes, but you would see so many people walking. And everyone was quite thin over the last 35 years. What's happened is that they've become quite a bit heavier. And we start to see obesity in countries like Bali and Vietnam, where it was just not a thing, except for someone with, a real genetic or hormonal disease. And people stop using their legs, and they aren't able to walk as far. And that's that kind of atrophy actually also happens in the brain. So the researchers in the study, they did it with students because, hey, it's MIT. They've got lots of students lying around. 54 student participants divided into 3 groups. They assessed them over 4 months. They had a ChatGPT LLM group. They used that exclusively for essay writing. They had a search engine group that used traditional web searches like Google and no AI at all. So they must have turned that off. And a third brain-only group who wrote the essays using only their memory and reasoning. And they wrote the essays based on SAT prompts, which, of course, when you're taking the SAT, you don't have anything but your brain in the room with you. And then in an additional surprise session, the participants switched. Some of the large language model users had to write without AI. Some of the brain-only participants were newly introduced to ChatGPT. And here are the key findings. Neural activity and connectivity was lower with AI use. The more AI used, the less engaged key brain networks were, especially those involved in memory, attention, and executive function. AI users retained less. Most ChatGPT users struggled to accurately quote from their own essays, unlike those who used only their brains or a traditional search. Sense of authorship dropped. AI users had a more mixed sense of authorships, with some participants claiming full ownership and others denying it. And satisfaction with their essays was mixed. For the AI group, consistently higher for the search engine groups. And the take-home lesson here, I think, is that a short-term benefit in productivity may actually lead to long-term costs in cognitive critical thinking, creativity, learning, and memory. And you could see those changes over 4 months in a group of highly educated students at MIT. So this is, imagine if you've got someone who retires and basically just watches TV. What's going to happen to their brain, especially with daytime TV? But let's assume that they're even watching something good, like movies or science podcasts. They're still not really using their brain. It's very passive, and you wonder whether they'll be able even to remember most of the content of what they see because they aren't actively working with the content. And I know if I'm trying to learn something and remember it, I have to make flashcards, or I have to underline it and then go back and reread the parts I've underlined, and then maybe write margin notes. God help you if you get one of my books after I've been through it, because it's marked up horror. Anyway, it helps me remember, so that's why I do it. I engage with the material. Human teachers were able to detect patterns of AI-generated content and tended to score them lower in originality and structure. Interestingly enough, AI-powered judges gave higher scores to the AI-generated essays. So there should be no surprise there. But cognitive challenge in learning tasks strengthens neural pathways, just like physical challenge strengthens motor pathways. It boosts memory, it builds critical thinking, and therefore, when people come in and they say, okay, I'm starting to have a few memory problems, what do you recommend, doc? What supplements should I take? And I say, you should take music lessons in an instrument you don't play, or you should learn a new language. You should make your brain do some heavy lifting. You should also make your muscles do some heavy lifting, by the way. About the only thing besides hard brain work that will help you build new synapses is hard muscle work, which measurably raises blood levels of something called brain-derived neurotrophic factor, BDNF. So thinking about thinking, which is essential to learning and self-awareness and studying, declines like crazy when the AI is doing all of the heavy lifting. Interestingly enough, the people who wrote their essays with AI had a hard time quoting from them, but they also had a hard time remembering what their essays said. So it isn't really yours if you use too much of this. I think it can be very helpful for repetitive tasks, for processing large amounts of stuff where when you're with a human, you zone out and get bored. The nice thing about AIs is, at least so far, that doesn't seem to be a problem for them. So let's let them get bored doing the boring task, and let's us do the interesting tasks. I got this wrong on a test recently, and a little background on this test. I am keeping my board certification with the American Board of Family Medicine, engaging in a relatively new pilot program that they've evolved. And what it is rather than having to take a board exam every 10 years where I go to a place and they're very creepy. It feels very carceral. Last time I went for one of these tests, they had a device that read the pattern of veins, or maybe it was arteries, in my hand, and then that had to match the previous time I'd come in. I guess they were worried that I'd switch out with someone in the bathroom who knew more about cardiology than I did or something. But no, I took the whole exam. But it's expensive, it's onerous, and it really doesn't test your working knowledge, and you don't learn anything. I think that's the most important thing. It's nice if you can learn things. I really like this new system. Every 3 months, I have to answer 25 questions and get a certain amount of them right. It's open book, but they're they're tough questions, and it really makes me, like we were saying just now with the Chat GPT, it makes me work with the information I have, or if I have to look something up, work with the information I've looked up. And I got this wrong on a test, and I was like, why did I get that wrong? My ego was immediately involved, and I went down, and they give you references, so I pulled the reference, and yeah, I got it wrong. It is new information, came out about a year before COVID, and there was a lot going on during COVID, so I think this one got quite the news that it would have. But one of the more popular and commonly used antibiotics, the fluoroquinolones, often used, for example, for community-acquired pneumonia, skin infections, and they're a really great drug. You can get bacterial resistance to them, but initially, of course, that wasn't the problem. Later on, we started seeing a disproportionately large number of Achilles tendon ruptures in people who had taken this antibiotic, and we investigated, and it turns out, yeah, it weakens cartilage, it weakens tendons. This whole class of drugs has a kind of toxic effect on the strings and ropes that hold your body together, and so we have to be careful in who we give it to. You certainly wouldn't want to give this to a competitive athlete or someone who played basketball or soccer for recreational purposes. It's so that one kind of got into all of our heads, and we've been careful and warned people to, cut back on their activities, etc. It's also associated with retinal detachment, which I guess makes sense because, again, that's collagen little fibrils that are holding the retina against the back of the eyeball. But this one really got my attention. Recently, they came out with a warning against aortic aneurysms, that fluoroquinolones can trigger the rupture of aortic aneurysms. And there are a lot of people out there walking around with either a preaneurysm or a dilated aorta. It's not quite reaching the level where we would put you through the risk of fixing it, because it's a big surgery to fix the aortic aneurysms. Instead, we say, well, it's under 4 centimeters, so just be aware if you develop a sudden ripping, tearing sensation in your back and a lot of abdominal pain, you should call 911. And now what we're finding is that you should also not ever let a doctor give you a fluoroquinolone. So out there, folks, I didn't know this. I'm a primary care doctor. Glad I'm taking these tests, because I would have just gotten the question wrong on the exam let's see, when would I take it? 2033. And by then I might have given quinolones to a few people. So it's the first I'd heard of it, and I am now even more motivated to do this quarterly exam. But also, I want to let everyone out there, anyone you know who has an aneurysm or a dilated aorta, history of hypertension and smoking, those are probably the people who should just have the doctor and the hospital put fluoroquinolones on their adverse drug reaction list. Nope, I haven't had an adverse drug reaction yet, but I don't want one, and I have that dilated aorta. So, I don't, but if I did, I would certainly stay away from these drugs. As it is, I stay away from these drugs because I have tendons, and I really don't want to rupture my Achilles tendon. So we're coming up on the end of the hour. I've gotten a lot of calls and questions over the last couple of years about the circulating tumor antigens as a diagnostic test. And there are tests that you can get that will look at the epigenetics. They'll look at the methylation pattern. They'll look for changes, microRNA. There's various different things you can look for that are only made by cancerous cells or precancerous cells. And, that's really, really wonderful and important, but there's a lot of false positives. But circulating tumor DNA is also in people who have residual disease. we need to study what we can do to prevent them from having an adverse event. So to prevent them from having a new cancer show up. And this study comes out, it was using a commercial test called Signatera in people with stage 3 colon cancer. And what they found in this study was that celecoxib, which is Celebrex, it's an anti-inflammatory drug related to ibuprofen. Actually, if you have circulating tumor DNA and you take celecoxib, it cuts your rate of recurrence in half. So, on the other hand, if you don't have it and you take the celecoxib, you don't get a benefit. So there's something about this anti-inflammatory drug that's somehow impairing the cancer's ability to thrive in its new metastatic condition. Because obviously, if there's residual disease anywhere, it's if it just sort of sits there and grows like a freckle, it's no problem. It's when it is able to become invasive. We don't know the mechanism for this, but we do know that regular people who take regular ibuprofen, perhaps for some other indication, actually have lower levels of colon cancer at baseline. So there is a protective effect of this, and it's not surprising, but it's nice that we can track who needs it. And I think this idea of looking for residual circulating tumor DNA and using that to see whether or not, if the levels are going up, then the cancer is growing. If the levels are staying static, then probably, yes, you've got a residual cancer somewhere, but it hasn't, it's in a holding pattern, which is the idea of just kind of, how shall I put this, arm wrestling our cancers to a draw. Got time for one last story. Interesting and surprising effect. We've always thought that inflammation was related to aging, that it's sort of invariable that we become more inflamed as we age. Turns out in the industrialized world, yep, that's true. But in some indigenous communities, it doesn't seem to be the case. This study looked at 4 groups, from all over the world, 3,000 adults from 4 countries, and it showed that chronic inflammation is a feature only of industrialized living. So the groups in question were, it was a study done in Singapore. The groups in question were the Semane from the Bolivian Amazon, the Orang Asli from peninsular Malaysia, and looking for the other groups that were, Italy and Singapore, both 2 very highly industrialized countries. And the studies measured levels of cytokines, those inflammatory compounds like IL-6, which we all heard a lot about during COVID. And these are inflammation signals. And they changed with each age group. High levels of cytokines were associated with age-related health problems, but what's interesting is that the people who were from the more primitive cultures that were living closer to the way we evolved to live, more of a hunter-gatherer type existence, actually did not develop inflammatory diseases of aging. I'm sure many of them, I don't know that the lifespan on the average of the people was higher, but if you lived to be old, you didn't get inflamed the way that the Westerners did. Well, that's about all for this week's podcast. Please go to AskDrDawn.com for news about our future plans. For now, this is Dr. Dawn saying so long and stay healthy.
Ask Dr. Dawn is brought to you by Jiva Media. Production and editing by Charles Bansky. Music by John Scoville.